Hi everyone, today we're going to be talking about metabolism in general and enzymes specifically. So, alright. Your metabolism, well, okay, so let's back up a little bit. Overall, your cells need energy. They need a constant input of energy, and you get energy from the food that you eat. The only organisms that don't have to eat are the primary producers, the ones that can do photosynthesis, like plants and algae. Okay? All the rest of us have to eat. We need that input of energy in order for our cells to work. Okay? So life is built on chemical reactions. You are a walking bundle of chemical reactions. And in those reactions, you're transforming energy from one form to another. Don't ever forget that law of thermodynamics. We can neither create nor destroy energy. So don't ever say that a reaction makes energy. It releases energy or it takes in energy because it is just transforming it. Okay? So all the energy on this planet comes from the sun. Our primary producers take in that solar energy and they convert it to organic molecules and ATP. Okay? And then herbivores eat those organic molecules and they use it to make ATP and their own organic molecules. And then the herbivores are, yes, this will make you sad, eaten by other things. And those organic molecules from the herbivore go into the carnivore or the predator and are used to make ATP and the carnivore's organic molecules. Okay? Your metabolism, we usually talk about metabolism just when we're talking about a person's ability to break down food. Oh, I have a really high metabolism. Metabolism is a lot more, though, than just having to deal with diet or food. It's every single chemical reaction that makes up you, through it from the second you are conceived until the moment you die. Every reaction you do as you build, as you break down, as you change, that is all your metabolism. Okay? So metabolism is going to involve forming bonds between molecules, taking two things and making them something bigger. Types of forming bond reactions are called dehydration synthesis, just plain synthesis, or also anabolic reactions. Anabolic reactions are building reactions. That's why they're called anabolic steroids, because those steroids help you build more muscle. They're building. They encourage building. Right? Metabolism is also going to involve the breaking of bonds between molecules, taking something big and breaking it into smaller pieces. We might call those, that might be a hydrolysis reaction. Hydrolysis is going to be water lysing, water splitting, water cutting. Okay? Or a digestion reaction. Or it's going to be also called a catabolic reaction. Okay, so anabolic building, catabolic breaking down. All right, here's some examples. So here's a dehydration synthesis. In dehydration synthesis, you've got two molecules that are going to come together. You're going to take a hydrogen off of one and OH off of the other. Those are going to come out together as water. So water's coming out, so that's dehydration. And the two molecules are going to move back together. Okay? Hydrolysis is the opposite. You've got something that's got to get broken down, and the way that's done is that water is shoved in there. A hydrogen goes on one side, and OH goes on the other. So water goes in, splitting with water, putting together by removing water. And you're always going to have enzymes that are going to make this happen. So here's another picture of that. Alright, so some chemical reactions are going to release energy. They're going to have an extra amount of energy that's going to come out. In biology, we call those reactions exergonic. In chemistry, you probably heard exothermic. Exergonic is basically the same thing, just a biology term. Sorry, we like to be confusing. This is going to happen when we digest polymers. Remember, most of your organic molecules are polymers, and we've got to break them down into smaller pieces. As you break them down, as you do hydrolysis, you're going to get an output of energy because it's becoming less organized, so you're going to a lower energy state. Some chemical reactions require an input of energy. These we call endergonic instead of endothermic, endergonic. Okay? These are going to be building polymers or dehydration synthesis, building up stuff. Okay, you're moving to a more organized state, taking little things and putting them together into more organized structures. That is a higher energy state, so it takes an energy input. All right, so let's look at some graphs of exergonic versus endergonic reactions. Okay, here's an exergonic reaction. On the side over here, you've got amount of energy. So you see your reactant has a higher energy state than your product. So as you go from reactant to product, energy is going to be released. What's this little bit here? Hopefully you remember that almost every reaction requires at least an initial input of energy. But as long as the output is higher than the input, then you still have energy released. So energy is released, that's a digestion reaction, for example. We measure the amount of energy released, and we call it delta G. Okay, so delta G is a change in free energy. Free energy is energy available to do work with. Okay, if energy is released, then it's a negative delta G. All right, an endergonic reaction is going to have an input of energy, because you see the reactants are at this low energy state and the products are at a higher energy state. So in order to get from low to high, you've got to put in energy. Okay, these are like synthesis reactions. When you're putting in energy, the delta G is positive. Your textbook has some kind of complicated reactions for calculating delta G. You do not need to worry about those. You just need to know that delta G is a measurement of energy, and that when energy is released from a reaction, it's negative. When energy has to be put into reaction, it's a positive delta G. Okay? All organisms require energy to live. They got to get energy from someplace, right? So your energy, yes, comes from the food that you eat, but how do we then use that energy? Well, we do it by coupling reactions that release energy with reactions that need energy. So this top reaction here, this is going to be a breakdown reaction, so it's going to release energy. Okay, so digestion releases energy. We're now going to take this energy and use it to make this synthesis reaction happen. Watch this. So the energy goes in, and it makes the reaction happen. Okay, so we put together reactions that release energy with reactions that need energy. Okay, something that kids always want to know. If a reaction is downhill, why doesn't it just happen on its own? Why aren't starch molecules just breaking down into glucose spontaneously all the time by themselves? Well, the reaction can happen on its own, but it happens very, 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 very slowly, and it's because of this hill right here. Okay? The covalent bonds that hold the starch together, that hold all those glucoses together, are really, really, really stable. And in order for them to break, they're going to have to be destabilized. In order to destabilize them, you've got to put in some energy. Okay? So breaking down large molecules requires an initial input of energy, a little bit of input. Okay? We call that input activation energy. And it's going to make the bonds that hold the biomolecules together less stable and make it easier for them to break. Okay? The activation energy that's required for most reactions that happen in your body, it's too much energy. You could, as they show here, you could put a match to your glucose and get it to break down, but then your body would be so hot that you'd start dying. So that would be a bad idea. All right? Too much energy to expose your living cells to. You can't just add heat to make the reaction happen. So instead, you've got to have something that can reduce that activation energy, make it easier to get over this little hill. Make it easier to get over the hill. And the thing that does that, they're called catalysts. They speed up a reaction by reducing the amount of energy needed to start the reaction. Okay? So there's the amount of energy needed for an uncatalyzed reaction. There's the amount of energy needed for an uncatalyzed reaction, or for the catalyzed reaction. And as our friend the penguin says, whew, takes a lot less energy. It's nice. All right, so what are the catalysts that cells are going to use? They're going to use a specific biological catalyst called enzymes. Remember, almost every enzyme in your body is made of proteins. Okay? How do they work? Well, most enzymes are proteins. There's a few that are made of RNA. They're going to help reactions happen. They're going to increase the rate of reaction without being consumed, and they do that by lowering the activation energy. They don't do anything to change the overall free energy. So the delta G does not change. It's just the activation, that initial hill that goes and is smaller. So the reaction happens faster. It's still the same reaction, it just happens faster. Okay? Almost every reaction in your body requires some sort of enzyme, so they are really, really specific. So that means you have thousands of different enzymes in your cells. So each enzyme does a different reaction. Since you have so many reactions, you have to have thousands of different enzymes. Okay? All right, so here's some vocab we want to make sure that we know about. The substrate is the reactant which is going to bind with the enzyme. It's the thing that actually needs to have the reaction happen to it. Okay? And then when the enzyme and the substrate bind together or form, it's not really a bond. When they come together, they form a temporary association, and we call that the enzyme-substrate complex. The product is what comes out of the reaction. And then the active site is the actual 3D spot within the enzyme that the substrate's going to move into and interact with. Okay, and it's a 3D space because all enzymes are proteins, so they have a big, huge 3D shape. So there's going to be a spot in that 3D shape where the substrate is going to fit. All right, some things to know about enzymes. They're reaction specific, so each enzyme works with usually one and only one substrate. Okay, the fit between the active site and the substrate is going to be because of hydrogen bonds, ionic bonds, some polarity, all sorts of things like that. Enzymes are never consumed in the reaction. They bring in a substrate, help the reaction happen, kick the products out. Bring in another one, help it happen, kick it out. And they can do it thousands of times a second. Thousands or more reactions per second. Thank goodness for enzymes. They make things happen quickly. Okay, and they are unaffected by the reactions. They just keep working and working and working and working and working. Okay, but they are going to be affected by cellular conditions. Because of the fact that enzymes are proteins, any factor that can affect a protein structure is going to also affect your enzymes. So that's going to be temperature, pH, salinity. Those are going to be the big ones that are going to really impact how your enzymes can work, because they're going to change the shape of the enzyme. Once the shape is changed, the substrate doesn't fit anymore, nothing works. All right, when we name enzymes, we usually name them for what they catalyze, and their names almost always end in an ASE. So if you see an ACE, you know you're looking at an enzyme, okay? And usually the first part has something to do with the reaction that they do. So sucrase breaks down sucrose, protease breaks down protein, lipase breaks down a lipid, okay? One of the only ones that doesn't follow that convention that you're ever going to see is pepsin. And pepsin was named and discovered before we came up with this convention, and for whatever reason they just never changed it. So pepsin breaks down proteins. It got the pep part from the polypeptide name for proteins. Okay, we used to talk about enzymes working like a lock and key model, and this was actually what was covered in my biology textbook when I was in high school. Yes, I'm old. But it's too simple of an idea. We used to think that the substrate fit into the enzyme's active site so specifically, so perfectly, that it was like a lock fitting into a key. But then we realized that it just was too easy. It wasn't really explaining what was really happening. Plus, because once you get that locked in there so tight, how is the substrate molecule supposed to change to do the reaction it needs to do? So with some more studying, yes, here's a slightly dirty joke from Miss Foglia. Gotta love her. What we learned from some more studying of enzymes is we actually came up with an idea called the induced fit model, which we really think is a much more accurate description of how enzymes work. And what this means is since the substrate and the active site are fitting together in a 3D way, that as the substrate comes in, the substrate and enzyme are going to change shape to help get everything in tighter and make the reaction more likely to happen. Okay, we call that a conformational change because it's a shape change. And it's going to bring the chemical groups in to a position that it's going to make the reaction easier to happen. And since the reaction happens easier, it requires less energy input, thereby lowering activation energy. The whole point. All right, so here's an example of how this works. So in a synthesis reaction, when the two molecules that need to be put together come into the active site, they come in and they're brought close together, and they're turned and twisted towards each other in such a way that the actual atoms that need to interact for the bond are right next to each other, and then it's much easier to put them together. And then the opposite is true then for digestion. If you've got a big molecule that's got to be broken apart, as that big molecule comes into the enzyme, it's going to be stressed and stretched in such a way that the actual bond that needs to be broken is going to be made weaker, and then takes less energy to actually break that bond. So both of these situations are going to make it so that less energy is needed to make the reaction happen, and that's the whole point of enzymes. Okay, so hopefully that all helps make enzymes make a lot more sense. I'll see you guys next time.